All right, so this is the second chapter in our biochemistry unit, and this is all about carbohydrates. First of all, the carbohydrates have a general formula. They are usually a specific number of carbons mixed with a specific number of water molecules, and depending on how many carbons there are, depends on how many water molecules there are, so on and so forth. So usually these types of chemicals have a composition of C something, H something, O something. So like for instance, C6H12O6. And so most carbohydrates have a various structure where these numbers can change to a different ratio. The carbohydrates are defined as sugars and polymers of sugars. So basically, remember, a polymer is just a really long chain. So one sugar or a various number of sugars. So another name for sugar is saccharide. And if we have a monosaccharide, we have a one sugar chain. If we have a disaccharide, we have a two sugar. If we have a polysaccharide, we can have 3+ sugars all in one carbohydrate. So let's talk about monosaccharides now. Remember, monosaccharides are our one sugar compounds. These are sugars with molecules that contain 3 to 6 carbons. So when we're talking about our formula for sugar, this number right here is going to be 3 to 6. These usually have one carbon that contains a carbonyl group, and a carbonyl group is a group that has a double bond with an oxygen in it. So like for instance, if we have a carbon chain of various lengths, remember monosaccharides are 3 to 6. Basically, we are going to have a carbon that contains a carbonyl group, which is a double bond oxygen, and then the other carbons will contain hydroxides coming off. And this group can be found at various places. And remember, all of the other bonds are hydrogens to make everything happy. So some examples of some monosaccharides, and this is a really big picture and it's hard for us to hone in and see it all. But if we notice, we have all of these double bond oxygens and we have these hydroxide compounds. So if you notice here, all our hydroxides, they are, I'll use the highlighter. We can see hydroxide, hydroxide, hydroxide. These are red. Okay. We also have our double bond oxygens, and these aren't as obvious to see, but if we look at this guy right here, we have a carbon. It says CHO, but what that means is we have a carbon. It's connected to another carbon over here. That's the rest of the chain. And then we have a double bond oxygen, and then we have a single bond of hydrogen. And a lot of times shorthand, it's written just as CHO, as you see right here. And so these are often called aldoses, and these are also the left-handed forms. Okay, and we'll talk about what left-handed means here in just a second. Okay, each of the sugars on the previous slide had at least one chiral center carbon. And chiral is a word you probably have never heard before, but what that means is that it is attached to at least 4 different groups. So chiral means that this carbon right here, so one of the central carbons, because remember, it can be a chain up to 6, so let's do one with 4. So maybe we're talking about this carbon right here. This one could be termed chiral, and what that means is that the 4 compounds coming off, one to the left, one to the right, one to the top, one to the bottom, are all going to be attached to different groups. And what that means is they are going to contain a mirror image. So like for instance, your left hand and your right hand are mirror images of each other. They're identical. They both have 5 fingers. They're probably approximately the same size. You probably even have similar wrinkles in your hands, but at the same time, they are mirror images. They're not the same thing. Every time you add a carbon to this chain, you actually double the number of isomers. So for instance, this one has, or if we look back at the previous slide, let's go backwards. This one, the top one right here, It only has one chiral center, so it's going to exist in two forms. It's going to exist as the left-handed form and the right-handed form. But as you add chains, so like this one over here, it exists in four different, because we're doubling. And then these are going to exist, and so it just kind of go down the line as to all the different ways you can rearrange it. Every carbon doubles the number of different possible choices that you're going to have. All right, so another type, so again, these were aldoses. We also have ketoses. These elements contain their double bond oxygen in the middle of their carbons. And so we were just looking at our aldoses right here, and our carbon double bond oxygen was located at the end. And all those examples I just showed you, we had that double bond at the end of the chain. Well, ketoses have the double bond in the center carbon. And again, these can be anywhere from 3 to 6 carbons long. And so as long as that double bond is not on carbon 1 or carbon 6, it is technically going to be called a ketose. We also have hexoses, and basically what these are, when we have our carbon chain, and remember our carbon chains have the end or the middle are going to have the double bond oxygen, but all the rest of them have the possibility of having these hydroxides attached to them. Well, what can happen in a hexose is these hydroxides are called alcohols, these OH groups, and basically what these do is these react with the carbonyl group, and what happens is they produce a hemiacetal, which basically means they're going to start to kind of wind and twist around each other until they form a cyclic structure, which is honestly a sugar, and it's a hexagon, so it has 6 sides because it contains 6 carbons, and so we call these hexoses for their 6 sides. Glucose is the most important. It is generated through photosynthesis and can be transformed into other sugars by different processes. So let's talk about disaccharides now. Monosaccharides are 1 sugar. Disaccharides are 2 sugars. So glucose was an example of 1 sugar. So now I'll start talking about 2 of them combined together. So 2 sugars fastened together. So one example would be sucrose. Sucrose is sugarcane, and what sucrose is it's technically a glucose molecule plus a fructose molecule, and they are combined together as we see over here. They're both a cyclic structure. This one is a hexagon, this one is a pentagon. These are both separate sugars. The left one's glucose, the right one's fructose, and they are bonded together right here in the middle with an oxygen. This is making sucrose for you. Lactose, which is the middle one, basically what that one is, again, it is glucose, which this time is written on the right, and it is being mixed with galactose, and this is actually what is found in milk or dairy products, and again, it is bonded in the middle by an oxygen. Maltose, which is the one on the bottom, is again a glucose, but this time it's a glucose plus a glucose molecule, and again, attached by an oxygen. Maltose is actually the product of starch breakdown in the brewing industry, so this is how we make a lot of our beers and other malt type beverages, which is why they're called malt type beverages. Okay, so some examples of our disaccharides are honey. Honey is 79.6% sugar and 17.2% water. How we create honey, or what bees do, is they break sucrose down apart into their glucose and fructose. Because if you remember, sucrose is made out of glucose and fructose, and it's only joined together by one bond. And so what the bees do is they break this oxygen bond, and then when they do that, they're going to get the separate parts, glucose and fructose. And so honey is actually 31% glucose, 38.2% fructose, 7.3% maltose, and then 1.3% sucrose. And so not all of the sucrose gets broken down, and we have a little bit left over. Since sunlight, which is technically energy, is needed to make sugar through photosynthesis, this energy doesn't just disappear. This energy is stored in the sugar, it is stored in the bonds. So when we break these bonds, when our body is breaking down the sugars into things that our body can use, it releases energy, and this is why when we eat different types of starches and sugars, we get a rush of energy, because as we break these bonds down, the energy has to go somewhere. Energy is always conserved. And so this leads us to something called metabolism. Basically what happens is our body cannot use disaccharides in that form. So what we do is we break them down into monosaccharides. Monosaccharides is what our bodies can use as to transform it into energy. And so in order to do this, we use enzymes, which if you remember from last chapter, enzymes are things that speed up chemical reactions. And so in this case, the enzymes actually speed up the breakdown of disaccharides into monosaccharides, which helps us consume our energy faster than it normally would. And so it's really easy to remember the enzyme versus amount during the sugar or the saccharide because their names usually go together. So like for instance, maltase is the enzyme used to break down maltose. Sucrase is the enzyme used to break down sucrose. And lactase is the enzyme used to break down lactose. And so it's really easy to remember which one goes with which because usually the enzyme and the sugar have the same name except for one letter, an A versus an O. Okay, so this is why people are lactose intolerant. If lactose is not consumed, so a lot of countries and a lot of different ethnic groups, they don't consume dairy or milk products after they are weaned in infancy. And so if milk or milk products are not consumed, lactase will no longer be produced. Your body will stop producing it because if you're not using it, why should your body produce it? And so what ends up happening is we see that 90% of Chinese and Japanese are lactose intolerant because it is not common for these groups to commonly ingest milk. And however, 20% of Europeans are lactose intolerant because it is very common in their culture to consume milk and dairy products. So, therefore, in countries where milk is not as prevalent or not available, we also have an increase in lactose intolerance. However, to fix this problem, you can take lactase, you can take this enzyme, usually in a form of a pill. You can take it when you're getting ready to eat any dairy type product and it will break it down for you so that your body can digest it. This is also why people who are lactose intolerant might have stomach issues or gassy or however you want to put it because the sugar breaks down or sugar just sits there because your body can't digest it because it can't digest disaccharides and it can't break it down either, and so it just kind of like ferments in your stomach, and so that's what produces all of the bad symptoms that has gone along with it. The next thing we're going to talk about is polysaccharides, and remember these are the very long chains, various lengths of chains of sugars. These are usually created from cyclic glucose, so again remember glucose exists in a hexagon shape and we call this cyclic. Basically, when we're talking about glucose, we have alpha D glucose, and that is the kind we're talking about starch. This produces to store excess glucose in plants. And so basically plants usually make more starch than they're going to need, and so they can then store it as in excess because if it's glucose and it's going to be used. And so they change it just a little bit and they make it a longer chain and they store it for future use. This is often found in tubers like potatoes. This is why potatoes are so starchy because honestly, they're just an abundance of starch waiting to be utilized, and so it's just being stored. Amylase actually breaks down the starch into maltose, and then we have more enzymes that can break it down into glucose. So it's actually like a two-step process. Glycogen is basically polymerized starches saved. And so when in our bodies, when we take all of this excess starch and we start to store it, we actually store it as glycogen. And basically what we do is we put a whole bunch of glucoses together to make a starch and we store it in that way. The beta form, which is beta D glucose, and if you notice, alpha goes with this symbol, alpha. Beta goes with this symbol, beta, and these are Greek letters. This is cellulose. Cellulose is used in plants and it is used as a structural material. This is what gives plants their ability to stand up straight and not just lay flat on the ground. It's this polysaccharide cellulose. This is not digestible by humans, and so when we're eating those parts of plants, it's hard for us to digest it, but at the same time it provides fiber, which is very important to our diet, and fiber has actually been shown to possibly decrease the chances of cancer, specifically colon cancer. And so even though it doesn't seem to be digested by us, it's at the same time doing some good. And so it still needs to be in our diet. Some other carbohydrates that deserve mentioning is chitin. This is a molecule that is very, very, very similar to cellulose. And basically what we use chitin for, or what organisms use chitin for, is they use it to make exoskeletons of invertebrates. So this is what insects have, or shrimp, or lobster. This is what protects them, because these don't have bones. And so they don't have any way to protect their organs. And so instead they have this exoskeleton which serves their protection. We also have hyaluronic acid. And basically what this is this is a lubricant and shock absorber in joints. And this is also can be found in bacterial cell walls to kind of give the ability to change shape without causing a lot of harm to your body. And then the last one is pectin. This is often found in cell walls of plants, and this is actually what is responsible for fruits forming gels. For instance, when you're making jelly, if you're making strawberry jelly or grape jelly, this carbohydrate pectin is what is actually responsible for causing it to gel up through that process. And the last thing I want to talk about are antigens. We talked about that a little bit in the last chapter. Antigens are what signals your body friend or foe. Antigens on invaders tell your body, oh, this is bad, and your immune system kicks in and it tries to kill it. Other antigens can tell your body that this is not a foreign object, this is supposed to be here, and so not to fight it off. So remember, those are what antigens are. And so basically sugars are what makes up the antigen markers on the outside of the cells. This is what tells your body this is good or this is bad. And these antigens can also be attached to proteins or lipids. And so this is how your body recognizes something that is invading it versus something that's supposed to be there, all by these antigens, which are made up by sugars.